In today's lesson, we will be introducing a pipette, a laboratory instrument, along with serial dilution and a 96 well plate reader. Without further ado, let's get started. This is a pipette. Pipettes are one of the most commonly used laboratory instruments and have the primary function of measuring and transferring accurate measurements of a liquid. Pipettes come in many different shapes and sizes. At our laboratory, we can use different pipettes to measure from 10 microliters all the way to 1,000 microliters. There are two key components of a pipette that you must be aware of: how to adjust the pipette so that it measures the desired amount of solution, and the two stopping positions. To adjust the pipette, spin the top of the pipette towards the right to increase the number of microliters absorbed. To decrease the amount of liquid drawn, spin it to the left. In terms of the stopping position, a pipette has two key stopping positions. The first stopping point is used to gather the desired amount of solution. The second stopping point is used to release the solution gathered inside of the pipette tip. To use a pipette, you must first take the pipette tip onto the pipette. For the second step, press the pipette to the first stopping point and insert it into the solution. Then, let go of the button and lift the pipette out of the solution. There should be liquid inside the pipette tip. To release the solution, press the top button all the way down and into the desired location, such as a 96 well plate. Congratulations! You now know how to use a pipette. Moving on to serial dilution. Serial dilution is another common laboratory technique used to dilute or decrease the concentration of a solution. In this video, we will be showing a simple serial dilution procedure for fluorescence, absorbance, and luminescence. First, fill one of the wells in the 96 well plate with the desired amount of the sample using a pipette. In this video, we are putting 100 microliters of our sample into the first well. Then, using a buffer solution, fill the desired number of wells with 100 microliters of the solution. We are using water for a buffer solution. It's important to note that you must change the pipette tip when using multiple solutions to prevent cross-contamination. Then, the diluting begins. Using a pipette, transfer the original stock solution into the next well plate that has the buffer solution in it. From there, mix the solution around by pushing and releasing the pipette button. Then, repeat the process until the desired number of wells is reached. To test the accuracy of your pipetting and diluting skills, you can use a 96 well plate reader. To use the 96 well plate reader, go to the back of the machine and turn the machine on. Then, the monitor must be turned on. From there, open the Glomax Discover app. In this video, we used a fluorescent solution, so in this scenario, please select the fluoresce option. Then, select the wells that you are trying to measure. This can be done by simply tapping on each well that you are using. Then, click the door button at the top to open the 96 well plate reader. Once the door is opened, insert the well plate inside. To close the door, click the door button once more. Then all you do is click start and the numbers will indicate the fluorescent levels. Thank you so much for taking this wild pipette and serial dilution journey with us.